Yeah, so my name is Sujay Champati. I am a rising senior and I'm from Pleasanton, California, so in the Bay Area. And this is my school mentor research presentation. My mentor is, as everyone knows by now, Aniruddha Chaturaj. And for this research project, I worked in the area of computational biology. So let's get started. So starting off with a brief introduction, we want to set the scene for this project. So in this project, our main focus is focusing on the activities that occur within cells. So first, what is a cell? A cell is the basic biological unit that all living things are composed of. So plants, you and me, we're all composed of cells. And the cell contains the ingredients for life. So think of it as a really small microscopic container. And these ingredients are floating around in this liquid inside that container. And in the average human, so you and me, there are approximately 37 trillion cells in our body. So, you know, that gives you an idea of how small of a setting we're working in. Now, when we talk about these ingredients of life, we're usually talking about biological molecules or biomolecules for short. So what are molecules? Molecules are basically very small structures that work together in groups to perform very specific biological tasks. For example, a group of molecules, including DNA and RNA polymerase, they work together to read a person's genetic code. A different group of molecules work on generating proteins based off this. A different group of molecules will work on, burning glucose, providing you with energy. And on the top right over here, let me move this down. On the top right over here, you can see a diagram of a cell. See, it's kind of like a container, and there are ingredients floating around in this container. But most importantly, on the outside, you can see molecules that are living within the cell, right? And some of them may be familiar to you, like glucose, insulin, DNA. Everyone's heard of that one, right? Now, again, molecules don't actually look like this. This is a representation of a molecule. On the bottom right over here is, on the bottom right over here is another representation of molecules. And if each of these, colored ribbons is a molecule, you can see how they interact with each other. And when they interact again, they form some larger structures that have certain biological functions. But what's important to understand, especially for this project, is that these interactions of molecules within cells have a certain element of stochasticity to them, which means that these interactions are random. For people who are having trouble conceptualizing this, think about the cell as like a really big lake, okay? And let's say I toss two magnets, which are two molecules, into the lake, right? Now, eventually, over weeks or months, these two magnets may come into contact and they may interact. They may repel each other, attract each other, something. But depending on the lake that I toss these magnets into, these magnets might meet at different times and they might have a different type of interaction. So that's what I mean. In every cell, for every molecule, there's a certain level of fluctuation of that molecule. Sometimes, if there's molecule A, let's say the baseline level for that molecule is 5 molecules within a cell, right? Sometimes that'll go to 7 molecules. Sometimes it'll drop to 3 molecules. It's going to be hovering around a baseline, but that level of fluctuation is called biological noise. Now, there are two types of noise. There's intrinsic noise and there's extrinsic noise. Intrinsic noise basically means that within one cell, how is the quantity of a molecule varying? So how is it going up and down, right? Extrinsic noise means cell to cell. So let's say I have a group of 100 cells, and at time equals 0, there's a certain level of, let's say, molecule A within each of these 100 cells. How is that quantity varying, right? So there's two types of biological noise. And over here on the bottom left, graphically speaking, you can see how a person can identify what the intrinsic noise and the extrinsic noise of the system is. So if our baseline over here is that gene A should be equal to gene B, right? So gene, let's just say for this project, that's a type of protein. If gene A and gene B are supposed to be at the same amount, the level of variation from this line of each of these single data points, that is intrinsic noise. Extrinsic noise means, are these data points, if everyone can look at this, if everyone could look at me, right? If all these data points are like bunched together, that means there's less fluctuation. The levels of gene A and gene B are really close together. That means there's less extrinsic noise. If they're really spread apart, then that means there's lots of fluctuation, right? So there's lots of, so there's more extrinsic noise. So if they're bunched together, there's low extrinsic noise. If they're spread apart, there's more extrinsic noise. And noise, again, just means fluctuation, right? Okay, so noise was the first important concept to understand for this project. And the second is biomolecular condensates. Okay, so these can be subunits of each cell that perform certain functions, and they are formed, and this is very important, when proteins or molecules, which we just talked about, when they form clusters within a cell. Okay, so when these molecules form groups, they're touching each other, they're interacting with one another. That's what a biomolecular condensate is. Now, the process by which biomolecular condensates are formed, the process by which molecules form clusters like this, is called liquid-liquid phase separation. Okay, now the figure on the bottom left here, which you saw before, illustrates this process. Okay, so suppose I have a cell that has none of molecule A in it, right? And I start adding molecule A. So now adding molecule A isn't as simple as like pouring molecule A in there, but let's just say it was. If I kept adding molecule A, the concentration, which means how much molecule A is there per unit volume, that's going to increase. And beyond this threshold concentration, what happens is there's going to be clusters, molecular clusters that start to form. And eventually what we get, these big blue droplets are molecular clusters, okay, clusters of molecule A. There's two phases that form, okay? One is a dilute phase. The dilute phase is when your molecules of A are kind of just floating around, right? They're not really interacting with one another, and they might be, maybe there's pairs of them, but that means it's again, by the name, dilute, right? So they're just floating around. And then the dense phase means there's clusters. There's large clusters of molecules that form. Now, that was with one molecule. If there are two molecules, there isn't a threshold concentration anymore. But based on research that my mentor has done, we know that there's a quantity called KSP, which you can see down here. Okay, KSP. This KSP dictates what that threshold is, okay? And the KSP is a numerical quantity, but it's nothing but the product. You multiply the number of free A's there are and the number of free B's there are, and then you get what's called the KSP, okay? Now, if this is sort of difficult to understand, I have an example over here. Many of you have probably poured salt into water. If you start pouring salt into water, the salt starts to dissolve, right? But if you start pouring more and more salt, what happens? The salt starts to collect at the bottom of the cup because you can't dissolve anymore in there, okay? So just like that, there's a threshold concentration beyond which no more is going to dissolve and they're going to form these large clusters, okay? So that's the basic premise of our project. Okay, so let's move on. Okay, so now the research question that we're attempting to answer relates to the two concepts that we just talked about. How does the noise or the fluctuation in protein counts change when a system exceeds its KSP and start to undergo that liquid-liquid phase separation process, which is just that clustering process, right? Now, in a paper written by Adam Closen, who's a leading biologist in 2020, just last year, he hypothesizes that liquid-liquid phase separation, this clustering, is a mechanism that helps reduce this fluctuation within cells. So based on that, what we've done is we've come up with two graphs over here, okay? So these two graphs are graphing the total concentration, which means as we keep on adding two molecules to a solution, what the total noise is going to be. So we think, so far, we think that beyond this threshold concentration, which we think is at about 140 micromolar, that this noise is going to go up. It's going to go up, and then at that threshold, it's going to plateau, or, and again, that threshold is where liquid-liquid phase separation happens, or it's going to go up. And then since, in the paper, it was proposed that it's a mechanism to reduce noise, it's going to go down. Okay. Now, if we look at this graph, if we look at this graph on the left, we can see that as our total concentration of A4 and B4 increases, that KSP value, that solubility product, which is what we call KSP, is going to increase and then plateau, right? Because again, if we think about salt, the dissolved salt after a point is going to remain the same, right? You can't dissolve any more salt. Similarly, you can't have any more free A4 and B4 after that threshold. So in figure B over here, if you see before the threshold at 80 micromolar, the cluster size is relatively small, right? You got your total molecules is 800 over here, 400. So we're increasing the number of molecules, but we're also increasing the volume to maintain the same concentration. So over here, we have A4 and B4. There's 80 micromolar, which means we haven't gotten that to that clustering point yet. And the main cluster size is just 1 to 10, right? Which means they're free floating. But after, at 160, which is 20 micromolar after 140, we can see that, okay, the clusters have started to form. We have some that are free floating, but we also have ones that are, sticking together, interacting, forming clusters. Okay, so how did we go about actually doing an experiment, right? Because we didn't have access to like a laboratory. So what we did, we used something called rule-based modeling. Okay, what rule-based modeling is that using this environment called virtual cell, what I did was I put a bunch of rules into this environment that just say, what is my reactant? What are my reactants? In this case, my reactants are A4 and B4, right? Because they're free floating. That now, what is my product? My product is a way in which A4 and B4 can interact. Okay, so if you look up here, this is my A4 and this is my B4. I call them A4 and B4 because they're 4 binding sites. A binding site is basically like a sticky place where something can come and stick and react. Okay, so if there's 4 binding sites for A and 4 binding sites for B, and each binding site can interact with all of the others from the other molecule, I'm going to have 16 different rules, right? So I defined 16 different rules, and I ran a simulation, which is just saying, suppose this happened once, based on the probabilities that these reactions are going to occur, because if 2 molecules are together, there's a chance they won't react. Based on the probability that they'll react, what are my levels of A4 and B4? And on the bottom left over here, if you look closely, these are the levels of the molecule, right? So if you notice, they're not going up and down a ton. They're hovering around that same level, but there is a fluctuation. And finally, using Python code, we take the data we get from a virtual cell and we say, how do we measure the noise? How do we measure the fluctuation? And second, how do we measure, these basic statistical values such as, mean, median, mode, things like that. And then lastly, how do we plot this data? So that's what the Python code did for me. So moving on to our results, the first thing we wanted to see is liquid-liquid phase separation even happening, right? And based on this trend, we see it is happening because again, that solubility product is going to plateau because there's no more salt that can be dissolved, right? I'm saying salt because, relating it back to the example, but there's no more protein that can be free floating. They all have to form clusters after this 140 mark, right? So it goes up, goes up, goes up, begins to plateau because that's why the solubility product remains the same. So this is the trend of the data. We know liquid-liquid phase separation is happening at around 140 micromolar. Okay. Now next is more related to our research question, right? The coefficient of variation we want to plot versus the total concentration. Now the coefficient of variation sounds complicated, but basically what it means is it measures fluctuation of protein counts. Okay. So again, proteins are molecules. How many are there in a cell and how does that vary, right? That's basically what we're trying to measure. And we proposed, again, if we go back, we hypothesized that that noise is either going to plateau or it's going to drop off after 140. But here, after 140, it doesn't look like that clustering or liquid-liquid phase separation helped anything or did anything, right? Because look, it just continues to increase. So when I saw this at first, I was pretty concerned. but as we will see soon, it is not a cause for concern. Let's go to the next one. If we look at each individual plot, so there's a lot of data on this page, but again, each of these individual plots gives you individual measurements for each concentration. So the x-axis is A4 concentration, the y-axis is B4 concentration, okay? And the total concentration is listed on the top. So we want to increase the total concentration and see how is A4 and B4, how is that noise, how is that fluctuation increasing, decreasing, or whatever it's doing, right? So total concentration 40 micromolar, we add more A4 and B4, we keep adding, right? And so this is the progression. Now, if we look at total noise, which we just looked at, which was coefficient of variation, you can see it's increasing, right? So total noise here increases, 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 and increases, right? So that's not, something that we wanted to see. And the intrinsic noise, now remember again, this is within one cell, and that's measured by how far these points are away from this line. Where do you see the largest fluctuation from the line? Here, they're kind of conforming to the line. And here, there's a lot of points. If you gave me that data without a line, I don't know about you, but I wouldn't really see much of a trend over there. They're varying a lot from the line, which means the variation within a cell is a lot when our concentration is increased. But let's look at the extrinsic noise, okay? So this is what kind of blew my mind the first time I saw it, because I thought my experiment had completely failed. But if we look at our extrinsic noise, we have 0.001 here. These are pretty arbitrary units. We're just trying to find a measure of extrinsic noise, okay, a measure of the fluctuation. Over here, it's 0.002. Now remember, at 140 micromolar, what happens? Liquid-liquid phase separation happens, which is the mechanism we're interested in, and see how noise changes from before to after. So let's see, 0.001, 0.002, 0.004, okay, moment of truth. All right, stays the same, right? 0.004, 0.004, and 0.004. Now, if you want to see that graphically, right, let's say, you don't believe me with all the numbers, you think I'm just making this up. We said how close together the data are means there's less extrinsic noise. How far away they are means more extrinsic noise, right? Now, beware of the scale over here, but where are most of the data points over here located? It's between about 11 and I would say like 15, right? What about over here? 11 and 15, right? Over here, again, about 11 and about 15, which means our noise cell to cell, means let's say we have 37 trillion cells, but let's just say we had 100. The variation between all of those is not changing after the liquid-liquid phase separation happens. So if you look at it over here, again, we can see our intrinsic noise, our total noise, and our extrinsic noise. And our extrinsic noise over here is the one that's plateauing. Total noise is increasing, the intrinsic noise is increasing, but the extrinsic noise is reaching a plateau, which, according to our hypothesis, with total noise is supposed to happen, right? Look how similar these graphs look. And I actually made this one before I even got this one, right? Pretty mind-blowing. Okay, now to our conclusion. Our conclusion statement says, liquid-liquid phase separation, or these clustering of molecules, provides a mechanism that puts a clamp on, which means causes to plateau, the extrinsic noise or cell-to-cell noise of this biological system that we model. Now, that's a pretty long conclusion statement, but what that means is the variation of those molecules between a bunch of cells that we have, it stays the same after liquid-liquid phase separation happens. So if we ask, do our results support our hypothesis? Kind of, because it doesn't support it with total noise. We thought that would happen with total noise. Total noise is just a straight line up, right? So we keep increasing. But our results do support our hypothesis in terms of extrinsic noise, right? Which means cell-to-cell variation plateaus when our concentration increases. Okay, real quickly, modifications in future projects. One of the things that I would like to have done is just add more molecules, do more simulations. That's just a matter of time. But another thing that I was thinking of doing and that my mentor suggested was maybe take an actual biological process like transcription, which is reading a genetic code, and see if the noise in that system is affected the same as this, arbitrary A4 and B4, right? So that's something that I think will be very interesting to experiment with. And finally, I'd like to thank a few people. Thank you to Miss Malhotra and the school mentor staff, because they provided this awesome program for me during COVID. And I want to thank my parents who supported me through this program and my education. So thanks, Mom, who's on this call. And a huge thanks to my mentor, Ani, for helping me through this entire project. I learned to say I don't know a lot when I was doing this project, because he would ask me a lot of questions and I had no idea what the answer was, but he helped me through it. And in the process, I learned so much. He was really patient. He's a really nice guy. And thank you so much for doing this. And thank you to everyone here who listened to my presentation. Thank you.